Hello and hi everyone. Today, me and my group mate from Group 4 are going to present about the discussion between the case study of microbial bioremediation as a robust process to mitigate pollutants of environmental concern versus our lab findings. Now, let me introduce me and my group mates. I'm Mahindran Ravindran. My matrix number is SB20001, and my group mate Harnita Harshini. Her matrix number is SB20009. Now, let's look at the introduction. This case study is about the huge environmental pollution caused by the regulated or uncontrolled release of waste material loaded with threatening, dangerous, and carcinogenic contaminants of rising concern have contributed to the flourishing industrial and biotechnological advancement and unrestricted growth. Several remediation procedures, such as chemical, physical, or a combination of two, have been developed and used to combat a variety of contaminants. Biological techniques, such as bioremediation in general and microbial bioremediation in particular, have gotten a lot of interest for pollution removal from various environmental matrices. This case also studies with appropriate instance current difficulties and restrictive boundaries associated to the implementation of genetically engineered bacteria as bioremediation factories on contaminated areas are highlighted.
Next, I'm going to talk about the discussion, which is the comparison between the experiment that has been carried out in the lab and also the about this case study. First, the experiment that has been carried out in the lab is to identify and evaluate which the two sample, the water sample from Sungai Gading and also Tasik Kuantan, is more heavily polluted and potentially dangerous to animals, aquatic species, and also human. There are 7 tests that has been carried out, which is BOD, COD, total suspended solids, pH measurement, total nitrogen analysis, ammoniacal nitrogen analysis, and also the last one is phosphorus analysis. Moreover, let's talk about the case study, which is the most of them necessitate that a significant degree of abrasion, aside from the unprecedented scale of the agricultural revolutions. Today's rapid industrialization methods have been carried out, which is to avoid the environmental pollution by releasing a slew of dangerous contaminants. There is a pressing need to successfully prevent, reduce, or degrade new environmental toxins to maintain a safer and healthier environment. Several remediation procedures have been developed and applied to a variety of harmful pollutants to found out that which has been dangerous to this environment. Traditional method used in the case study. First of all, biological, physical, and chemical approaches, as well as a combination of these methods, are the conventional use methods. From this case study, as I observed that there are no conventional method used, but mostly the author focus on microbial bioremediation to eliminate the environmental pollutants. But in my suggestion, for some reason of the stability, ease of maintenance, and reliability, physicochemical treatment or ion exchange or adsorption can be extensively utilized. Moreover, the physicochemical removal of pollutants using activated silica powder also can be activated by the carbon and also precipitated calcium carbonate in coagulation and flocculation methods.
Now, let's look at the suggestion of bioremediation method for the case study. The method that we have chosen to suggest is bioventing. Bioventing entails improving bioremediation by increasing the activity of indigenous bacteria by controlling airflow and giving oxygen to the unsaturated zone. Nutrients and moisture are added to assist bioremediation with the ultimate goal of achieving microbial transformation of contaminants to a harmless condition. This bioventing is popular among other in situ bioremediation strategies, particularly for repairing places that have been damaged by light-spilled petroleum compounds. As we look into the picture, we can see the process of bioventing where the airflow is given from underneath the ground surface with the help of low-rate air injection and blower. And when the airflow flows from bottom to top, the contaminated soil is given by airflow and giving oxygen to the unsaturated zone. That is all about the discussion on the case study and our lab findings. Thank you very much for watching.